years ago. Of course, they disappear already. So how to study? So the story is the scientists go to Alaska. Alaska is a very cold zone in the Arctic zone in United States. So of course they also have quite many people died due to Spanish flu in 1988 in Alaska, and therefore they try to find a dead body in that area because it is a very cold area. So even people died and go to the tomb, the body is still not rot. So by using this way, then they found that we rescue the 1918 influenza virus from a fake woman. I think because the fate and dead woman, of course, because the fate can somehow protect the quality of the virus. Anyway, they rescue and they identify one thing very interesting, and it is because they validate that 1918 influenza virus cause human death very quickly, maybe just in 1 day or 2 days. It's because this virus can cause extremely high level of human immunity. And this immunity will cause the fever and the swelling and the body fluid leaking into the lung. So actually, all the patients die due to they have no oxygen. So it's good to know, but it is also a very bad story because which means global warming might be forced loss of ancient virus from the permanent frozen lands to our real society. Because, for example, they can even rescue the virus from 100 years ago. So how many virus still residing in Russia? I don't know, in a very cold area. So anyway, no matter how many story I told you, the fact is at that time people still don't understand what was the pathogen. They just know they are not bacteria. Because viruses are very small, of course they cannot see. So the milestone for biology, there are some very important steps. One is the discovery or invention of electron microscope in 1930s. At least at that time, people can eventually understand, this filterable liquid has something look like this, but at least they still don't understand. And in 1950s to 60s, the Watson and Crick's they discovered that DNA is the inheritable genetic material. And people start to realize, these particles also contain some of these filterable stuff also contain DNA. So it seems they are some genetic material. Of course, the later on people discover that. So the people eventually realize this filterable stuff is genetic material. So of course they can transmit to each other. And maybe the most important thing for biology study is the in vitro cell culture system. And at that time, people can eventually culture mammalian cells in culture dish. So therefore allow people to study the mechanism of virus at the cell level. So after this important milestone, in the and scientists use maybe 20 to 30 years eventually identify this terrible human disease caused by which kind of virus, and this virus genome belong to which family. So biology actually starts far later, nearly 100 years later than bacteria. So still quite many things haven't been solved. And this one is the global outbreak of the pandemic disease. Theoretically, no matter which country you are, if you have any of the case belong to the global notice infectious disease, you need to report even one case. And if you are interested, you can search this website. You will see what thing is outbreak in your country. I think so far everything is COVID-19, or maybe not so interesting. So here just let you know why even one virus infectious disease, you still need to report to WHO. It's quite simple because now have COVID-19 outbreak, so everyone should understand something. For example, if your classmate just beside you just be confirmed COVID-19 positive, yeah. And we all know virus have some incubation time, approximately 3 to 5 days. And this incubation time, then he or she have no symptom, but he or she can transmit the virus to the others. So why only one case still need to report? Because if your country have one case, usually means your country already have 10,000 cases. Understand? So that is the power of the virus disease. One case usually already means 10,000. Yeah, happen in the next week. So need to be well controlled. Quarantine. So anyway, we talk so many about how bad the virus are. This one is the life expectancy of human beings from 1970s to 2015. So believe it or not, if you just born in 1980s, you can only live 35 years old. Yeah, quite bad. But currently, we know that human average life is about 70 years old. So what happened here? We really need to thank microbiologists because most of the life extent depends on microbiology study. But if we point out some important time point, you will find something strange. For example, Louis Pasteur discovery that bacteria cause human disease in 1960s is here. And Robert Koch identified many bacteria pathogens cause human disease here. Invention of penicillin by British people is in 1928, is here. But actually, the application of penicillin actually in 1942 during World War I, because you discover doesn't mean you can mass produce. So what happened here? Actually, we have weapon over here, but people already live more than 30 years within this period. Of course, public health and epidemiological study also important, but actually, the thing that contribute the most is vaccination. And most of the disease are virus disease. So it's quite interesting. At that time, scientists actually didn't know what are the viruses, but they already invent some strategies to against virus disease called vaccination. I think this story everyone should have a brief understand. Anyway, the British medical doctor called Edward Jenner, he discovered that the ladies who collect the milks, called the milk ladies, and this kind of population is quite difficult to get the disease called smallpox. So it means if he hypothesized that a human, if infected by the mild and similar disease, might be able to protect from a very deadly disease. So at that time, he performed an experiment by injecting the cowpox into the young and innocent boys, and therefore already infected these young and innocent boys with real smallpox. And this boy, of course, survived. So that's why people have smallpox now. And believe it or not, smallpox, if you just live 100, 200 years ago, before 30 years old, you have 30% die due to smallpox. So that's why people live life expectancy so low. And if you have a more, and I think you also know that how Spanish conquered the American continent, mostly because they bring smallpox to America. And most of America, especially Indian, totally have no immunity for smallpox, so nearly all people die. So that's why only 1,000 Spanish can conquer such big land. So anyway, so let's see what happened after the smallpox thing. So every year in UK, in London. And around average 5,000 people per million died due to smallpox in London City. And after the vaccination start of the smallpox in 1980s, you can see that it nearly dropped to zero. So quite interesting because this thing happened even earlier than Louis Pasteur. So of course, how the people therefore invent the vaccination. So Louis Pasteur, of course, he identified they identified quite many disease might be caused by either bacteria or virus. Of course, at that time they don't know what is virus. So he tried to consult to follow the heterogeneous strategy, whether we are able to make the animals or human infected by some pathogens with similar symptoms but much mild symptoms, and therefore allow these vaccinated individual against the stronger pathogens. So what he performed is, and if you remember at the beginning, I told about some a virus called rabies. Rabies actually can cause 100% death rate if you don't treat the proper drug within 48 hours after infection. Once the virus goes to central nervous system, you die. Yeah. So anyway, it is a very deadly pathogen. So he want to create something mild. So how he create? So it is the story. So of course, Louis Pasteur don't know rabies is a virus, but it seems this dog had infected by rabies. Very very toxic. So he tried to collect some biomaterials from these crazy dogs and they inject into rabbits. Of course, at the beginning, rabbits have not very significant effect because anyway, these two species too different. But after repeat and repeat of this experiment, and Louis Pasteur eventually developed a rabbits like virus that's suitable to infect rabbit. So many so rabbit. Anyway, so anyway, it causes adaptation. So for example, COVID-19 at the beginning is actually in bad, therefore they adapt in human beings. So anyway, he tried to make a rabies adapt into the rabbit. And therefore, he found that this rabbit adapted rabies cannot cause severe disease or only very mild disease in dog. And therefore, he tried to see whether this dog vaccinated with this rabies can protect from real rabies. And the reality is yes. So he succeeded. So this technique called the adaptive mucin or the life attenuated vaccine. We use this one quite frequently. For example, measles vaccine is this one, mumps vaccine is this one, polio vaccine is this one, and what else? German measles also this one. Usually this vaccine is suitable for the very, very deadly virus. So interesting. So people don't know what is virus, but they can invent vaccine. So, and if you still remember the smallpox, actually smallpox is already officially eradicated from the globe because we just remember this FNDV story. You can only prove your country is FNDV negative if every single swine without vaccination but still have no disease. So smallpox is the same story. Actually, we hadn't vaccinated for smallpox for more than 30 years now, but currently not a single case of smallpox can be reported. So there is no smallpox virus anymore. Wrong. Actually, there are two. One is in American CDC and one is in Russia CDC. In the laboratory, biosafety level, all laboratory, they still keep smallpox. So when Russia get crazy, people need to be very afraid to do something crazy. So anyway, so many scientists also ask U.S. and Russia to how to say to discard these the final two vials of smallpox virus, but they reject. They say they need to keep this one in case this virus came out again when we still have something to research. Yeah, I think what they say is also correct. So anyway, the other virus that are going to be eradicated called poliovirus. Poliovirus can cause paralysis if the young children be infected. So it's actually a very highly transmitted disease and can cause lifelong problem because one leg will be paralyzed. So you guys are very lucky. Uh, so just see how many cases per year when there's no vaccine. So anyway, there are lots and lots of deadly pathogens. For the viruses, the very deadly one has been solved by the vaccination strategy, smallpox, for example, and some bacteria can be killed by antibiotics. So most of the so believe it or not, now you can live from 30 years old to nearly 60 years old. Actually depends on the microbiology study. Uh, so so now this question is so after 60 years old, of course we want to live longer. So if we want to live longer, we need to deal with one thing called cancer. Actually, cancer is a aging disease, of course, aging disease. So the fact is, if you live long enough, if any one person live long enough, you will definitely get at least one type of cancer before you die. Of course, if you are very lucky, you don't have. But if you live long enough, theoretically, you will definitely get cancer. Because cancer is genetic, how to say, it's the cells acquiring too many genetic material mutation abnormality, and this genetic abnormality and mutation accumulated according to the time and the environment you expose. So you can see that most of the cancer case reside in the population who are elder than 55 years old. So it's you need to confess it is aging disease. So if you want to live longer than 60 years old, you need to conquer this disease. Uh, uh, and believe it or not, 20% of the cancer, at least 20% of the cancer caused by virus. So let's tell you how the virus cause cancer or tumor. Actually, this kind of thing also happen in animal quite frequently. So just before 100 years ago, there's a doctor for Francis Drayton Royce, and he's a clinical doctor, and he has a friend, and his friend, for whatever reason, make a turkey as a pet. Anyway, so at one day, his friend come to his clinic and say, "Oh, doctor, say my turkey have a tumor." So of course, because he is clinical doctor, so they used to cut them off. Okay, so they remove the tumor, and just notice the time. This time is also the period people crazily find the so-called pathogenic reason for all kinds of disease. So yeah, this doctor is very lucky because he tried to answer whether this tumor is caused by certain kind of pathogens. So he tried to do all the things cough can do, and therefore he think, not bacteria. Okay, not bacteria. So what is that? So at that time, so you see, it is very lucky. Some very important scientific outbreak usually need many things together. So at that time, he read that there's some people who study plants validated that something filterable can cause disease. So he tried. And he tried to filter the smash of this tumor from the turkey and try to inject in the new turkey. Have a tumor again, and repeat. Have a tumor again. So he was the first one validate this filterable pathogenic stuff can cause tumor in turkey. in Turkey, not human. Okay. So anyway, it's a very, very long story. So according to his achievement, actually he should won Nobel Prize very early. But actually, he got Nobel Prize 60 years after he reported his study. So if you want to get Nobel Prize, you need to live very long. So why? Because of course during the pathogen finding period, tumor definitely or cancer definitely is one subject people want to crazily find what pathogens cause tumor. But unfortunately, during the same period, another scientist identified that the nematode, this is a parasite, might cause the gastric carcinoma in rat. Nearly the same time. Because this person is more famous, so eventually this person get Nobel Prize, and therefore he died. And therefore, after 1 year of he died, people discovered that the mice have the rats have gastric carcinoma because they lack of vitamin A, but not because the infection of this one. So of course, due to this reason, nobody believed his research during the same time. Everyone said, see this example. So he was very unlucky, but fortunately he lived very, very long. So he reached the milestone of biology. So let's repeat again: electron microscope, DNA discovery, in vitro cell culture system, and RNA discovery. So he eventually validated that the thing that can cause tumor in Turkey are an RNA virus, an RNA virus and can cause tumor. And the other thing is the first human tumor virus eventually be identified called Epstein-Barr virus that can cause lymphoma. So anyway, he eventually get Nobel Prize when he is going to die. Yeah. And this virus is extremely famous. This virus actually won 3 Nobel Prize. It is the first one. The first one is the identification of virus that can cause tumor or tumor virus. And the second story is because this RSV is an RNA virus, actually the scientists discovered that RSV infected cells carry the RSV genome as DNA. So they identify something called reverse transcriptase. And this enzyme only resides in the retrovirus. And the third Nobel Prize is two scientists called Ramos and Bishop, and asked one very important question: why RSV can transform the infected cell? Transform means make them into tumor-like cells. And therefore, they found that actually there's another virus, its genome very similar to RSV called ALV. You can see that their genome are nearly identical, but the RSV have a very small fragment called SOC, and additional to ALV, and therefore they studied this SOC. They found only these SOC genes enough to transform the cells. So their achievement is the identification of tumor oncogenes. So only this virus won 3 Nobel Medical Prize. So far there are how many viruses can cause human cancers? Let's go through. Hepatitis B virus, Nobel Prize, hepatitis C virus, and Nobel Prize last year, human papilloma virus, Nobel Prize 2008. And there's another virus, quite rare case, and mostly in the Northeast Asia, especially Japan and Korea, and can cause lymphoma. And the only one bacteria cause cancer, H. pylori, Nobel Prize. And there are still 2 major viruses still can cause human malignancies, but still have no vaccine and drug. One called Kaposi sarcoma herpes virus, and the other one called Epstein-Barr virus. And if you are able to solve the problem, I think you will also win Nobel Prize. Good luck. So here's just one story here. And there are some scientists tell us, discuss, there are too many kinds of malignancies. Every single one carry different mutation. How can we really conquer the tumor? Maybe we don't know how to invent the drug for every single kinds of malignancies because it takes lots of resource. But I think people already know how to prevent from cancers. For example, you just try to keep away from smoking, and actually you will reduce 33% of tumor incidence. And if you control your body weight very well, you will further reduce 20% of tumor incidence. If you don't know some trash food, 5% further. If you do sport every day, 5% further. Reduce sun exposure, 2% further. So if you can do these 6 things, actually you are already keep away from 60% to 70% of cancer incidents. I think you can have 10 years more life. So if you take a look of this table very carefully, actually you will find there's one thing you cannot control is the cancers caused by pathogens, especially viruses. So if you, by doing research for these viruses and find a suitable drug or vaccine against them, and theoretically we are able to eradicate these kind of human cancers in the soon future. So that is why nearly all such kind of research won Nobel Prize have reason because it totally eradicates this kind of cancer. So after the history, let's go back to basic science. So I think I already mentioned enough story, so I need to go back to reality, science. So let's go back to the basic biology. So we'll talk about the concept of virus classification, genome life cycle transmission, and other information. Yeah. So the difference, so maybe we still go through the 3 types of major microbial. One is, of course, bacteria, the other is fungi, and third one is virus. So the lifestyle is the bacteria and the fungi, they can totally live by themselves if you give them sufficient nutrition. Actually, you really think bacteria like to live inside the human body? I don't think so. Yeah. So, so they can totally live independently. But the virus totally need to depend on the host cells. And for the treatment, and no matter bacteria or fungi, you can find a suitable antibiotic to kill it because they're anyway different from mammalian cells. For virus, you cannot use antibiotic. And even for the drugs, you cannot easily find drug. Why? Because the virus live inside the infected cells. Of course, you can find many drugs that can kill virus, but you can also kill the cells. So that is why finding the drug killing virus is far more difficult than finding the drugs for bacteria and fungi. Because for the viruses, we also need to worry about whether this drug kill the cells of our own cell. And for immune response, bacteria and fungi mostly depends on innate immunity. And for virus, it definitely requires adaptive immunity. So it requires NK, T, and B cells. Even And here's the lifestyle for human beings. It seems there are less and less people right now. I don't know why, but some countries still have many babies. And the limitation is money, house, and bacteria. They use cell division, 1 to 2, 2 to 4, blah blah blah. And the limitation is no nutrition. For virus, the replication like this, here called replication or multiplication. So you can see that one single virus infect these cells, they can produce huge amount of viral particle. And the limitation is no cells to be killed. So usually virus cause very fast and dead disease. And here's the classification of the virus. And of course, also like bacteria, there are plenty kinds of different virus. According to their genome structure, we already mentioned the virus is either DNA or RNA virus, right? So for DNA virus, you can also separate them into double-strand DNA linear structure, double-strand DNA circular structure, or single-strand DNA linear structure. And for RNA virus, you can separate into double-strand RNA, single-strand RNA, positive strand, minor strand, single-strand RNA virus, and the segmented virus. And according to the most outside layer, whether they have a most outside layer composite with lipid bilayer, they will call the amylovirus. If they are not just caught by the capsid layer or the most outside layer, they will call the non-amylovirus. So basically, if you separate the virus by with oil without envelope plus their genome, they should have 1, 2, 3, 4, 14 kinds of family virus. And of course, and even if they are the same kind of category, for example, positive strain RNA virus envelope, and according to the genome orientation or the viral protein it can exhibit, they can be further separated. So there are thousands or the 10,000 different kinds of virus family. So it is a very brief category. So here all the viruses I shown here are the virus family that are documented in the medical biology textbook for undergraduate. So the thing I want to tell you is you just need to feel it. And there are many different kinds of virus that can infect human beings, and all of them have different genome and different shape. And the wavelength without envelope. This one is without envelope. So the most outside layer is capsid. It's a shape of protein, so it's very stable. And this one is the envelope virus with a lipid bilayer. The most outside layer is glycoprotein. And these kind of viruses are quite resistant, quite sensitive to detergent because their most outside layer are lipid. So why people why you need to at least understand the virus not currently is outbreaking belong to capsid or non-capsid virus, envelope or non-envelope virus because they have totally different physical meaning. So here's the life cycle of the virus. On the left side is the envelope virus. Right side is the capsid virus, non-envelope virus. So for the envelope virus, because the most outer layer is also lipid, so they have a chance to enter the cells called cell fusion. Because our cells most outside layer is also lipid bilayer. And the non-envelope virus that enter the cells called receptor-mediated endocytosis. Anyway, the cell will not die. And the different thing is how they release, because the envelope virus, if they want to go out, they can use one thing called body, because anyway, their envelope and the plasma membrane are all lipid, so out. But the non-envelope virus, the only way they go out is killing the cells, because they have no lipid. So usually the virus here, the disease speed are faster, but doesn't mean they are more severe. They just replicate faster and have symptoms faster because they will kill lots of cells. But here, it doesn't mean they necessarily need to kill the host cells. The host cells will eventually die, but not as fast as the non-envelope one. So here's just some summary of the physical and chemical character of the virus with and without envelope. Of course, the envelope virus, they're afraid from detergent, and they're afraid from the heat, the drying, or whatever. And non-envelope virus is they are stable. And transmission, the envelope virus, because they are very sensitive to dry out, so they definitely need to live in a moisture environment. So most of the things are transmitted through blood or aerosol or body fluid. And the non-envelope, they can transmit in all kind of way you can imagine. They can also transmit through oral fecal pathway. Means from shit, yeah. And